So I just want to welcome everyone and thank you for coming today. This is today's SCI webinar, which is part of a series of webinars produced by the ACRM and the ACRM SCI International Special Interest Group to help bring needed information to rehabilitation researchers and providers. So you are invited. Please feel free to join our SCI-ISIG. This is a link to join, and this is our web page, and you can connect with Denise Fife, our spinal cord injury ISIG chair, if you'd like to learn more information or if you'd like to join. So now I would like to introduce our speaker today, who's Dr. Ceren Yarar-Fisher, speaking on how microbiome research can change spinal cord injury care. But before we get started with the presentation, I do want to advise that this meeting is being recorded. I'd like to ask all attendees to keep your phones or mics muted and to keep your cameras turned off throughout the presentation. We do expect to have about 20 minutes available for discussion at the end of the webinar. So if you have questions, please type them into the chat feature and we will address as many of those as we can during the latter part of the hour. So I'm just going to pass the slides over to Dr. Ceren Yarar-Fisher, and it's my pleasure to have her here today.
Awesome, thank you. I will share my screen now.
Sorry, I'd just like to introduce you in a bit more depth first.
Sure, yeah.
Thank you. So Dr. Ceren Yarar-Fisher is the Vice Chair of Research in the Department of Physical Medicine and Rehabilitation. She's the Director of Clinical Research at the Bedford Center for Spinal Cord Injury and Associate Professor of Neuroscience and Physical Medicine and Rehabilitation at The Ohio State University. Previously, she was an Associate Professor at the Department of Physical Medicine and Rehabilitation of the University of Alabama. Her translational research laboratory focuses on understanding the pathophysiology of traumatic spinal cord injury in the acute and chronic stages, aiming at developing novel dietary and rehabilitation strategies to improve neurorecovery, metabolism, and microbiome composition. As such, Dr. Yarar-Fisher's lab is currently developing and testing therapeutic diets and electrical stimulation programs to evaluate new strategies to prevent neuronal death, promote recovery and function in acute SCI stages, improve body composition, gut microbiome composition, and skeletal muscle, metabolic, and bowel health at SCI chronic stages. She has led and collaborated on many projects, resulting in numerous peer-reviewed articles and book chapters. She's received several NIH and NIDILRR funding to support her research in SCI, nutrition, and microbiome. She was the program director and site-specific project PI for the UAB SCI model systems from 2016 to 2022. Her contribution to high-quality, well-funded research has been recognized both institutionally and internationally. The Junior Chamber of International selected her as one of the 10 outstanding young persons of Turkey for her work in academic leadership and/or accomplishment. She served 2 years as treasurer, chair-elect, and chair in the American Congress of Rehabilitation Medicine in the SCI-ISIG. She has recently been selected as recipient of the ACRM Fellowship. She's had the privilege of mentoring various students and trainees, including high school students, medical students, physical medicine and rehabilitation residents, PT students, doctoral students, and postdoc trainees. The students she has mentored are recognized for their research achievements via prestigious SCI research funding and academic scholarships. And without further ado, again, I'd like to introduce Dr. Yarar-Fisher.
Thank you so much, Hope. This was really great. Let me share my screen. Hopefully I'm doing the right thing. All right. Can everybody see my screen? Okay, perfect. Thank you for the nice introduction. You saved me time to go through everything you said. So I was going to talk about our laboratory, but as Hope mentioned, I was at UAB, started my career there as a postdoc, and then moved to a faculty position there. And recently, well, relatively recently, moved to Ohio State and started the ProSci Lab. And ProSci Lab, we have a website now, and I'm kind of invested in the lab website more than before because we hopefully will have community input in the website But if you want to join the website, it's the prosci.science. It shows our studies. There are some blog posts. It also talks about spinal cord injury care more in depth. So in Ohio, when I took the position, I was really lucky to work with a great group of people. And the reason I actually chose Ohio was because the microbiome science was really getting more improved, and we have better technologies in Ohio State to study microbiome. So that was really the decision maker for me. Phil, Dana, Jan, and I specifically focus on spinal cord injury from a chronic disease perspective. So we are really interested in understanding how organ systems work after spinal cord injury and how they're related to each other. Phil and Dana have done many research in animal models, neurorecovery, and metabolism, and Jan really worked on inflammation within the last 10 years, and I brought my metabolism, metabolic health, and microbiome expertise from humans into the team. And one thing I want to tell you about the Belford Center is that we started a repository for microbiome there. So hopefully it will become a national repository for everyone. We will share data there. We will share genomics analysis, data sets, and other things that everybody can use and maybe take some lessons or kind of design their own study. So it's going to be a very transparent science that we are planning to do in Belford Center. And if you have been to NIH meetings within the last several years, I think we met in 2019 or 2020, I can't remember, but we discussed how to set out goals for spinal cord injury research for the next 10 years. And the idea was that, and we all know this, there are a lot of investments done on neuroprotection for basic science trials, clinical trials, but we really don't have one drug that worked for everything, and it's impossible to do it anyways. It takes translations, 50 years of translational time. But I think the point is we need to really focus on different areas a little bit. We need to open our horizon to study different organ systems if you want to improve quality of life in spinal cord injury. And we have published these studies a long time ago. We are still publishing with new papers, at least the metabolic health part. But what we have shown over the years, and this has been reproduced by other labs too, there is this hidden obesity in spinal cord injury. It happens very quickly after injury, and it continues over time. It comes with inflammation. It comes with metabolic impairment. So if your patients or your participants are tested for fasting glucose and insulin, it may show you healthy ranges. So you wouldn't be really suspecting metabolic dysfunction. But what we have seen, again, over the years, if you challenge the system with glucose, if you do glucose loading in people with spinal cord injury, you will see that they cannot really utilize glucose for so many reasons: for autonomic nervous dysfunction, nervous system dysfunction, skeletal muscle extreme loss, and hormonal regulation. And now we think microbiome too. So when you look at the insulin sensitivity, people with chronic spinal cord injuries have always significantly lower insulin sensitivity. And if you look at injury levels, this is more prominent in females with tetraplegia, and this has been reproduced by us again. So it's not only one study. The tetraplegic women have the worst metabolic dysfunction, again, when challenged with glucose loading into the system. So by the time we were really interested in metabolic function, Phil's lab and some labs from Turkey started publishing microbiome composition changes in spinal cord injury. This happened it started from 2017 up to now, at this point. What they have shown in their studies is that gut dysbiosis, which we kind of define as all the beneficial communal bacteria, is replaced by pathogenic bacteria after spinal cord injury. This change very quickly after spinal cord injury impairs neurorecovery. That's one of the strongest data sets that I have seen at that time. The neurorecovery was significantly impaired by gut dysbiosis. They also show some metabolic impairments. They specifically look at the intestinal inflammation and capacity of nutrient harvesting within the intestine, which is incredibly important for metabolic function. So these were all impaired. And SCI microbiome science was new at that time, but the microbiome science is not new for anyone. Diabetes, cancer, neurodegenerative disease, this has been studied for years and years. We were just waking up to the microbiome impact in spinal cord injury at that time. So we really had a gut feeling that there is something to study there, especially for our interest for metabolic health. And 6 years ago now, Neidler funded a nutrition trial, and Craig Nielsen also funded a longitudinal trial at the same time, just looking at microbiome. So we were able to actually collect a lot of samples longitudinally, and then also test an interventional microbiome, which I will be showing you the results. But that was the perfect time, I think, to get into this area. So what I'm going to do the rest of the presentation is to show you what microbiome is a little bit, 101, very simple explanation, just so if you're not familiar with the microbiome science, you can familiarize yourself. I'm going to talk about very fundamental studies that change our mind about microbiome and metabolic health. I also want to show our results, our studies, and also show you the newest data that come from metagenomic analysis, which will be published hopefully soon, that you can actually have the paper in front of you. And then I'm going to talk about a little bit of future directions, what should be done for microbiome and SCI science. So when I start the lectures, I always want to go back to the beginning, the very beginning, where the planets are born. And the point out that the first and most successful organisms on the planet are microbes. They are bacteria. They have been around the longest. They are the most successful. They are also the most numerous. Everywhere we look on this planet, we can find microbes. They have evolved and adapted to live in the most extreme environments. This could be anything or everything from an acid mine to the incredible pressures at the bottom of the ocean. Sorry, I cannot change. Okay. And if an alien landed on Earth, this is a joke I always do with my lab, they would not think you're actually humans. For them, you're superorganisms. We, as well as other biological entity on this planet, have evolved in a microbial soup. In fact, we haven't just evolved in a microbial soup, but we co-evolve with the microbes. They live in and on us. We actually rely on them for functions that we ourselves do not encode in the human genome. And when you look at the right panel here, there is this bacterial data on the skin, and on the right side, there is the molecular data. And this is done by 16S analysis and the metabolomics on the right side. But what this is really basically showing you is that the red dots are where bacteria is the highest, and blue is the lowest. But even in the lowest abundance sites, the molecular data is very rich, which means bacteria still produces molecules that can impact the function of skin or other metabolic systems in the body. And this is just a lower abundance site. Skin is a very lower abundance size for microbiome. We bury the most burden in our guts. There are microbes obviously in the mouth and the whole way down through the GI tract, and they differ at different sites along the GI tract. And we think that's because of the prevailing conditions differ. If you think about the stomach, the pH is very low. In the lower gastrointestinal tract, which is gut, there is really low oxygen. So there are really strong selective pressures that drive the types of organisms that like to thrive in these distinct places throughout the gastrointestinal system. And when you look at the studies, I always have this question about, is the distal gut the answer for everything? We are like, it's not. You know, there will be a different bacteria in stomach, but we know that the abundance burden is mostly in the gut or lower gastrointestinal tract. So when you think about microbiome science, you will see papers looking at microbiome with different methods. For example, we take the microbes out of their environment, grow them under feast conditions in laboratory media, and studied what they do. So this has been kind of how it started, right? But now it's not enough for us because there are many organisms we cannot culture because we don't know how to grow them. We don't know what they eat. We don't know what they subsist on. And we turn on to molecular tools. And in my laboratory for the last 5 years, we really used 16S ribosomal RNA gene analysis. And this is actually most of the labs, I would say 80% of the labs so far have used to understand the composition of the microbiome. But the problem is we don't have high resolution in 16S. We really cannot go into species level. We also don't know the functions of the microbiome. We can see who is there and how to compare to other people, but we cannot really tell you what those microbiomes do actually as a function. So then we turn into shotgun metagenomics, where we can sequence the whole genome, and then we can understand the functions of microbiome. And it has a great resolution for species level analysis. And we also do metatranscriptomics and metabolomics. You can actually look at what microbiome is transcribing of the genome. We have never done metaproteomics, but it's a very heavily used method right now to look at the protein responses from the certain microbial groups. Another really amazing thing is to think about how much our microbiome overshadows our human genome. This is one study of European, Asian, and US populations, and they collect just over 1,200 fecal samples, and they examine it using shotgun metagenomics. So looking at all of the microbes and all of the genes encoded by the microbiome in those 1,200 fecal samples, what's staggering is that almost 10 million microbial genes were found across those 1,200 fecal samples. I want to let you think about that for a second. That is incredible, because this is an ancillary microbial genome that we carry around with us. These genes are not silent, they are actively expressed, and we rely on these genes for things like digesting our dietary components, metabolizing our drugs, and informing and influencing our immune response. So these are an important part of our physiology, an important part of what makes us healthy or diseased. Another fundamental study that I love and to discuss in presentations, the twin studies. Researchers found pairs of human twins in which one was obese and the other one was lean. And they collected feces of the obese individuals and the lean individuals, and they transferred them to germ-free mice. And germ-free mice are the mice with no existing microbiome. They're bred to lack a microbiome, and they're wonderful vessels for studying chronic disease. What these studies have shown us is that transfer of the obese microbiome into a germ-free mouse sets up an pathogenic microbiome, which is obesity-tended microbiome. Those animals gain weight a lot faster than the animals that receive the lean microbiome. So this suggests that the phenotype of the disease can be transferred from the patient by transferring the gut microbiome of that patient to a mouse. And that's pretty incredible. That means that the microbiome is responsible in large part of the obesity in this case. And what's exciting is that it's not only obesity, and this is another fundamental study that I really enjoy reading still to this date. They do human fecal matter transplants from a lean, healthy donor, improved insulin sensitivity in people with metabolic syndrome. They actually show incredible responses in insulin sensitivity and glucose disappearance rate when they receive microbiome from lean and healthy individuals. Again, this piece of evidence strongly suggests that there is a causal effect of the gut microbiome on host insulin resistance. If you are a fan of Fight Club, which I am, always remember this from this presentation too. What happens in the gut does not stay in the gut. It actually enters the circulation, and these small molecules, perhaps the little inklings, even microbes themselves, can translocate to different parts of the body and change the physiology of the organs, and they contribute to the health or disease of these remote organs. What's really exciting is that now we're seeing conditions like obesity, diabetes, Alzheimer's, and Parkinson's, cancer are related to microbiome changes, related to microbiome composition. So what happens to gut microbiome, or what happens to whole gut health after spinal cord injury? Sympathetic nerve fibers providing autonomic input into the enteric nervous system originate from the thoracic region of the spinal cord. whereas visceral sensory afferents carrying feedback from the gut snaps with spinal cord neurons that eventually transit to the brain. And another point is afferent vagal fibers also report to the brain, specifically informing of the conditions of the intestinal environment. So when you think about it, after spinal cord injury, there's an interruption or loss of control over these various pathways, and feedback loops pushes the intrinsic enteric nervous system away from the homeostasis and causes an autonomic imbalance. And what we think is that this autonomic imbalance impairs the gut integrity. So then the pathogenic bacteria, or again, the metabolites the bacteria produces can leak into the system, impacting other organs. And to support this idea, the first study that ever was published in humans was again in 2016 or 17. They show incredibly important changes in gut microbiome in spinal cord injury, and compared this to healthy controls. They actually did something interesting. They divided the groups into upper motor neuron syndrome, lower motor neuron syndrome, and healthy controls. What they have shown is that the SCI patients have a reduction of beneficial bitrate-producing bacteria. And I'm going to talk about bitrate a little later, but the idea is that bitrate has a lot of function in terms of metabolic health, intestinal integrity, and also inflammation within the whole system. There are diabetes that's showing that bitrate-producing bacteria actually could be a causal effect on insulin resistance in type 2 diabetes. So they kind of show this first time that there is a change after spinal cord injury in the gut microbiome, and it looks different than healthy microbiome. And there are some features linked to metabolic health and inflammation in the paralyzed microbiome. Another fundamental SCI study that we have seen in the literature is from Phil Popovich's lab. They use a thoracic 9 contusion SCI model, and they reported that as soon as a couple days after injury, the gut permeability actually decreases. There is translocation of microbiome from the gut into the system, and they have showed that bacteroides and clostridiales changes. Bacteroides goes down and clostridiales goes up. Again, these two bacterial groups are related to neurorecovery, related to metabolic health, related to inflammation, kind of mimicking what has been shown in the human study. They also show there are certain bacterial groups like enteroplasmates, clostridiales. They are inversely correlated with neurorecovery or motor function. And they also reproduced this data with different cohorts with different injury levels, showing that neurorecovery is impaired with the microbial dysbiosis right after injury. So our first study was done in people with spinal cord injury and compared their microbiome with healthy age and sex match controls. This was the first study actually ever studied the acute SCI human microbiome. What we looked at was that how chronic SCI looks different than acute SCI, and how they're together looks different than the healthy controls. And what you show, what you're seeing on the right side is a principal component analysis, which shows you how the data is clustered when you look at it as a group. So the different colors indicates different things. The blue is acute SCI, black is the chronic SCI, and red is the able-bodied controls. What you clearly see here, other than looking at millions of numbers in your data set, is that those groups behave differently. They're clustered differently, so their microbiome composition sits differently compared to each other. In this study, I don't want to bore you with lots of, because microbiome data is tables and tables of data and abundances of bacteria. But if I want to summarize what we have shown in this study, is that there is a significant difference in gut microbiome, and we measure it with beta and alpha diversity in individuals with acute SCI, chronic SCI, and able-bodied individuals. So if we cluster all SCI groups together compared to healthy controls, they have features linked to metabolic syndrome. And you can see certain bacteria groups that are related to this function. Inflammation-related bowel disorders, depressive disorders, which was another area that has been studied by animal labs for a while now. And they're related to antibiotics use. And definitely you would expect that because both chronic and acute SCI use lots of antibiotics. And also the chronic SCI group shared features linked to physical inactivity, which was really expected looking at physical deconditioning in the chronic SCI. We don't know this is whether SCI alone or antibiotics used together cause these results, but it's hard to actually separate this in SCI studies because it's a clinical practice. So we cannot really encourage our individuals or participants to stop using antibiotics. So I think this is the clinical reality, and we have to study around it or with it. This is what we have been doing in our latest studies. So our second study looked at, now we know there's a change in microbiome composition, even in acutely. Our acute patients were 4 to 7 days after injury. So, okay, this changes, and we think there's a correlation between metabolic health and microbiome. We wanted to see, just to focus on spinal cord injured individuals, if we phenotype them into metabolically impaired and compared them with metabolically healthy group, are there differences? Are their microbiome different? This was a very interesting study. I think it was a very important question. Even within the microbiome SCI group, we started seeing different changes in the microbiome, if they're metabolically healthy, if they're metabolically unhealthy. So again, you see a principal component analysis of people with type 2 diabetes or prediabetes. Both groups have spinal cord injury. And then the red circles will tell you the glucose tolerant group, which means metabolically healthy group. So there were clear differences in both groups. We show different type of bacteria increased in the diabetes group, like Microbiome, and certain type of bacteria decrease in the diabetes group, such as Firmicutes or Clostridiales. And again, this group that is decreased have function in butyrate production, and butyrate is heavily related to inflammation and heavily related to metabolic health. We are kind of mimicking the animal data and the first published human data in our own studies. So another important result from this study was we did metabolomics analysis. So we looked at small molecules in the system and correlated it with the microbiome composition. So what we have shown is that we found that phenylacetylglutamine and indoxyl sulfate was really high in the diabetic SCI group. And indoxyl sulfate is the end product of microbial fermentation of the amino acid tryptophan to indole, followed by endogenous oxidation and sulfate conjugation in the liver. So indoxyl sulfate in the literature, specifically in cardiovascular and hypertension literature, shown to promote vascular endothelial damage. And this damage is always followed by the inflammation markers being increased. And we went back to our inflammation markers, and then we showed that actually our higher inflammation was correlated with higher indoxyl sulfate levels in our data set. The second metabolite we found, these are gut-derived metabolites. So gut has important function in producing these metabolites. The second metabolite we found was phenylacetylglutamine, and phenylacetylglutamine results from liver glutamine conjugation of phenylacetic acid, which is almost exclusively derived from the microbial conversion of phenylaniline. So again, phenylacetylglutamine have incredible presence in the cardiovascular and hypertension literature. It increases actually blood clots. This is the biggest impact that's shown in studies. And this was high in people with diabetes and prediabetes in the SCI group. So the third study, this was a nutrition study funded by Neidler. This was a 5-year study. We looked at, so this study had many layers to it. We really were interested in, as a primary outcome, the metabolic outcomes. Can we improve metabolism or metabolic function in people with diabetes or prediabetes? And what we have shown, which multiple papers published, you can find those papers, definitely with a low carbohydrate, high protein diet with plenty of fiber, you can improve metabolic function. You can actually reverse insulin resistance into a healthy state. So our pre-diabetic or in the diabetes range group, they weren't using any medications. Their insulin resistance become insulin sensitivity at the end of the trial. So that was a very successful controlled trial in our opinion. And we were really interested to see if this healthy diet can improve the microbiome profiles in these people. And these were all chronic spinal cord injury. So what we have shown is that, again, the healthy diet with plenty of fiber were able to improve the composition. We have shown microbial communities that help with the community stability, alleviate intestinal inflammation, maintain gut barrier integrity, which was really important for us because the biggest problem is when you think about it, the bacteria translocates into the system. That's what we want to stop in the future. That's one of our goals. It promotes mucus production and it modulates immune responses. So these bacteria increase after a dietary intervention. So also, we have shown the bacteria abundances, they're implicated in inflammation, in metabolic disorders, and cardiovascular disease decreased in the group that received the healthy diet. And you always love when you reproduce your results again in other studies and then make a physiological impact or impactful claim. This happened with phenylacetylglutamine. We, again, as you remember, we show phenylacetylglutamine was really high in people with SCI and metabolic impairment. Now, what we have shown is that people who receive this diet, because the target was people with metabolic impairment, who receive this diet not only improve their microbiome composition, their metabolic health, but the gut-derived metabolite, which is phenylacetylglutamine, that is really important for cardiovascular health, was decreased in the diet group, which was, I think, very impactful for us to see that a 8-week diet intervention can reduce the gut-derived metabolite, which kind of tells us that the microbial composition or the bacteria who produces that metabolite decrease in the gut by following this 8-week low carbohydrate, high protein diet. Okay, this is the last data set. This is very fresh data, very exciting data, hopefully coming in physiologenomics very soon. But what we have shown here is that we did 16S ribosomal RNA analysis all along upon this point. But we were able to see what is in the gut and how it's different from different populations or different health profiles. In this study, what we did was we actually sequenced the whole genome of the bacteria. And then we wanted to see how are the functions are related to the metabolic health of individuals with spinal cord injury. This was funded by Craig Nielsen. It's a longitudinal study from 46 people with chronic spinal cord injury. Again, we looked at all the metabolic indices, such as OGTT, lipid panels, inflammation, and we correlate that with the composition and this time function of microbiome. And this is kind of a heavy slide. It's a heavy slide for anyone looking at a paper or looking at a screen, but this color map is really showing you the relationship with different bacterial compositions and functions to metabolic health indices, again, such as lipids, such as insulin sensitivity, glucose tolerance. In this paper, we identified functional pathways, and metagenomics analysis allow us to do that. We identified pathways and microbes that are correlated with metabolic health. They're very consistent with evidence from existing literature, existing metagenomics data sets. Specifically, microbes related to complex carbohydrate degradation and maintenance of gut integrity are implicated in metabolic health. That's what we have shown. While harmful microbial metabolites and gut inflammation are linked with worse metabolic health. Again, we always hypothesized that from our 16S analysis, but we did not really look at functional pathways ever before. And this cross-sectional analysis using MetaG workflow kind of showed our capacity to perform complex microbiome data analysis using multivariate data analysis approaches. And when you think about the bottleneck of informatics in omics science, it kind of limits the studies to tell us the big pictures. Because we can find metabolic disorders, we can find disorders in the microbiome, but connecting them with omics and multivariate analysis is a really hard art, in my opinion. And most probably, if you're doing microbiome research, you're kind of in the same problem with us. The bioinformatics will be really important in the future for microbiome science to move forward. Okay, so what's the big picture? I'm going to stop here after this slide so we can talk about it if you want to talk more about the specifics of the studies, but I want to give you the big picture now about what we understood from microbiome work that has been done over the years, and also what we have understood from our SCI and microbiome work so far. So now I think here too, we have an understanding that we have this ancillary microbial genome that influences our health. We carry millions of microbiome on our skin and in our guts that actually can translocate into different organs or different parts of the body, impacts human physiology. Microbiome expands the genetic and functional capacity of the human host. What happens in the gut definitely does not stay in the gut. Gut dysbiosis post-SCI is very clear now. We know it from animal to human models. This adversely affects host physiology, and significant differences exist still to the state from acute injury to chronic injury and to healthy controls. We still don't have a healthy microbiome profile we can actually compare our studies with, because microbiome science really doesn't allow that. It's very individualized, but I think you need to kind of start considering what looks healthy for your disease population with functions and composition of microbiome. Microbial-derived metabolites, in our case, indoxyl sulfate, phenylacetylglutamine, can influence metabolic function or may influence it. And microbes and functions related to complex carbohydrate degradation and maintenance of gut integrity are implicated in better metabolic health. This is all SCI study based. And the harmful microbial metabolites and gut inflammation are linked with worse metabolic health. So that's what we learned from our data sets. And a healthy, low carbohydrate, high protein diet with plenty of fiber can improve microbiome composition and improve actually the gut-derived dangerous metabolites that could cause cardiovascular problems in the future. So as the last point, what we should think about for microbiome science and SCI in the future. So the fecal transplant therapy, again, all these fundamental studies that I really like to read and look at is how people use this information as a clinical translation. So fecal transplants have been done, for so many years, especially for C. difficile. When I look at the C. difficile literature, the antibiotics are effective as much as 60%, but the fecal transplants is effective about 98%, which is incredible. So we can actually change the whole disease progression by transplanting fecal matter. And I mean, if you heard about the yellow soup for the soul, this comes from Chinese medicine. They used to grind poop and literally make a soup out of it to correct diseases. It's the same principle, fecal transplants. And what about bacteriophage therapy? Bacterial viruses are called phages or bacteriophages. They only attack bacteria. Phages are harmless to people, animals, and plants. So can we use bacteriophage therapy for improving immunity? We don't have to use a lot of antibiotics. We don't have to actually sweep the good bacteria from the microbiome. We can target certain pathogenic bacteria by using the bacteriophage therapy. So it's actually becoming a heavily used method for neurodegenerative diseases and cancer. Genetically modified bacteria. Can we modify bacteria to get microbiome do different things? For example, if there's a high inflammation in the intestines, can we target a certain bacteria that can release anti-inflammatory properties into the system? Again, this science is kind of being used for chronic diseases already, but can we carry this into spinal cord injury science? What about diet and microbial cocktails? Can we target certain bacteria? Because every time we give community talks and show our data or scientific talks in different places, at the end, everybody asks, are you building a probiotic or prebiotic or a symbiotic? I think what we don't understand, or I didn't understand as a novice scientist in microbiome years ago, the target of bacteria is really hard. You producing a probiotic for a certain microbiome is very hard. Most studies use very mainstream probiotic, or they use like over-the-counter probiotics, and they never show like incredible results. I think the reason is we don't know exactly what's going on yet. We did, 4 or 5 studies. We're still scratching the surface. We need more data. We need more data sets from different people, different injury levels, for us to ever come up with a targeted probiotic or a prebiotic or symbiotic therapy. So it's not really that easy. I never actually want to claim our studies will lead to a certain probiotic. Hopefully it will within the next 10 years. But again, as a cautious scientist, I can tell you that we need a lot more data. We need a lot more centers to contribute to the repositories and other things. We need data sets like NIH Human Microbiome Project, where they collected millions of samples from different disease populations, elderly, kids, and adults. So an SCI is very low, numbers-wise, very low population. So we may open our doors to international collaborations and other things. So these are the things to think about, I think, when you think about microbiome science and SCI. So most of the work I show you here, actually all of it was done at UAB, and my lab changed right now. I have still some members to the state in my lab, but then these were the people who worked in the SCI studies at UAB. So I'm thankful for the team members. Definitely, we couldn't do all this work without the team members, specifically my postdoc. Now she's the scientist in my program. Dr. Jali did most of the bioinformatics for this work, and it's one of the hardest parts to do. Also, we were funded by NIDLR, NIH, and Craig Nielsen for this work. So I'm forever grateful for the funding agencies for allowing us to do this work. And UAB Metabolomics and Microbiome Center have tremendous effort in these studies, because when we started, we did not even know how to collect fecal samples correctly. So we started learning from sample collection to study designs to bioinformatics, now metagenomics. So it's an ongoing learning process for us. And I think that's it. I can stop here and we can talk about it.
Hey, so I just like to thank Dr. Yarar-Fisher for a wonderful presentation. Very interesting. I always learn so much. I learned a lot more than I learned in September 2022 when I saw you speak last. So lots of interesting new information. And I just like to go to, there is a question in the chat from Jean. And the question is, given what's currently known, what actions do you recommend clinicians and people with SCI consider taking now to promote or restore gut health? For example, should people with SCI who have been treated with antibiotics for UTI consider having their microbiome evaluated to determine if therapy is warranted?
Yeah, I think that's a great question. It's a very multi-layered question. So the first answer is, I don't think we can suggest anyone any type of therapy yet. But what I know is that a healthy diet, so that's what we are seeing more and more. We have a new study going on. A healthy diet, which means you have plenty of vegetables, you have plenty of grains and a fiber. Fiber is really important, right? So a natural way of improving the microbiome composition. So what I would say to our patients too, if they're going through a big antibiotic kick, like if they're using antibiotics for any type of infection in their body, probably their good microbiome is already swept. And it takes about, I would say, we don't know if they're ever coming back, but it takes about that normal regulation happens within the next 6 months or so after using antibiotics. So whoever uses antibiotics, I think, should consider probiotic approaches, either eating more fiber or eating healthy diet, or any probiotic supplement that are suggested by their physician could be really helpful while they're using antibiotics. So this is one way they can maybe make that microbiome a little healthier compared to what was it before or during antibiotics usage. We just need more data. We can't prescribe anything yet. We just need more data from different labs, different profiles of people.
Thank you. So we have another question in the chat. So Jean says, a question for when it's time. Given what is currently known, what actions do you recommend clinicians and people with SCI consider taking now to promote or restore gut health? Oh, sorry. She said, thank you. That was a repeat of the same question.
No, that's okay. Yeah, there are labs doing exercise studies with microbiome, not an SCI, but anything I think we know. So all the organ systems are very connected to each other, and this is not our science. This is a science for the last 50 years or so. Anything I think healthy for like moving, eating healthy, and everything else that is healthy for us will be good for microbiome. This is at least what we are seeing in the data sets. I think the metabolic health has so many outcomes that impact other systems. So we always look at metabolic health, a measure of insulin sensitivity. Now we learn over the years also looking at inflammation and inflammation impact on gut microbiome, and now neurodegenerative diseases. So everything is connected. I think any healthy behavior, my opinion or my estimation is, will impact gut microbiome in a healthy way.
Thank you. So I do have a question myself. So you did a wonderful job of painting a picture from the animal twin studies and taking us through to the crystal ball. And I was just curious about those approaches you discussed in the crystal ball. Which of those, in your opinion, do you think is most promising and why?
So I'm really excited about the bacteriophage therapy. So we, in Belford Center, we always look at antibiotic use and other outcomes in humans and animals too. We really don't know long impact of antibiotics. It's bad in the short term. It's lifesaver for infections, but again, it's kind of sweeping a lot of things. If bacteriophages become a thing for SCI, instead of using broad spectrum antibiotics that kills many things other than the bad things, bacteriophage, it would be more targeted approach in the acute stages, specifically acute stages of injury. And other thing I'm excited about, the fecal transplant. So at UAB, I watched this happen actually on patients with obesity, with metabolic disease, with C. difficile, and the GI. I think the physicians are very interested in also trying fecal transplants for so many things. I think the skill set is coming to different institutions, different hospitals now for fecal transplants. The problem with SCI is that the data is very limited for us to target. It's not diabetes, obesity, cancer. You have millions of data sets or data points from different studies. We are really young in this. So for us to say something about fecal transplant is really kind of going against the science, making indirect connections to future data. So I think I would feel more comfortable for a bacteriophage, for example, if you're able to collect a lot more data to repositories, multi-site studies. But we need to remember also for multi-site studies, the geography will change microbiome. Microbiome is impacted for so many things. So we have to have really good analytical models that control for geography and other things. And these are, again, bottleneck in microbiome science. Even for NIH microbiome project, combining all this data from different geographical locations probably was a headache for them to controlling for these all variables. But back to your question, bacteriophage is what I'm excited about.
Thank you. And we do have one more question here in the chat. So in one of your results, insulin sensitivity was improved. So Srijana is just curious, did the research team get a chance to see for how long it was improved, or was that reversible after a certain time?
Very great question. We wish we could do this over like longitudinal fashion to see when it's reversed. We did not, we didn't have the funding on the time for that at that time. We had anecdotal evidence saying like those people who went through the diet calling us a couple months later saying, you know, they do probably like always pinprick in the mornings just to look at their values and things. So there were a couple of people call for saying that their values are still on board, but the point is, again, there's a confounder that they continue the diet. So we fed them very controlled way, which means we prepared everything in the metabolic kitchen, and then that was delivered to their home. We did food logs, and everything was kind of like controlled in a way. So after the study, they wanted the recipe. So we did a nutrition book for them and a recipe book and a shopping book. So some of them continue the diet, some of them continue the lifestyle. And these are the people who called us and say it's still working. But I'm not sure if it's like a longitudinal effect of our diet, or is it because they stick to the diet still and going as a behavioral intervention. And some of them called, just ask for protein bars. We served on, they loved it, and other things. But back to your question, I don't know. I think the ones who called us for improvement actually kept that lifestyle.
That's really interesting. So maybe another future research study there.
Yeah, that's what we're trying to do now. It's actually like at the end of the day, if you look at like the impact of your work, at the end of the day, maybe you will say, who cares if you fed someone with a healthy diet and they improved something? I don't think that way, that harsh, but at the end of the day, also, we need to make this a behavioral intervention, right, for people. If we are doing this work to show that this works, then how we can actually get people to adapt themselves to a healthy lifestyle. And spinal cord injury have lots of limitations already for shopping, for cooking, for other things. So how we can make this information more valuable and effective for them so they can kind of keep going that way is our kind of next goal from our nutrition studies.
Okay, and I don't see any other questions. So I'd just like to thank everyone for attending this talk, and thank Dr. Yarar-Fisher for providing another interesting presentation for our SCI-ISIG group. And just let you know, all of these recordings will be available in the next 24 to 48 hours. And please recommend this webinar series to your colleagues. Thank you.
Thank you.
Thank you so much. And thank you for joining. Good to see everyone.
Good to see you. We'll
See
see
you
everyone
later.
at 8.